Well, what I'd like to do is to now turn our attention to the development of other kinds of properties in the visual cortex that probably relate to the emergence and the construction of circuits that are intrinsic to the cortex itself, not just the topographic mapping of thalamus into the middle layers of the visual cortex. So I'll remind you that one important function of this cortical circuitry is to compute new functional properties. So not just to amplify the pre-existing properties that are inherited from the thalamus, but to actually construct circuitry in the cortex that can perform this important function of computation. Now, we talked about a couple of examples of computational properties that seem to arise as much from the operations of intrinsic cortical circuits as does the convergence of inputs from the thalamus. And two of those properties that I'd like to talk about now in the second part of this tutorial are orientation preference and direction preference. So I'll just remind you of what these properties are. Again, the discovery of these properties goes back to the seminal work of David Hubel and Torsten Wiesel. They were rightly awarded the Nobel Prize in 1981 in part for these discoveries. And what they did for the first time was to insert microelectrodes into the visual portions of the brain and record the action potentials from individual neurons. And they were the first to explore systematically the receptive field properties of the neurons that they encountered. And what they discovered when making a penetration in the vertical direction, such as this, is that all the neurons across the depth of the cortex that they could record from all shared similar receptive field properties. That is, they tended to represent overlapping regions of the visual world, and when they tested different orientations or different angles of bar stimuli, what they found is that these neurons all shared the same orientation preference. For example, in this illustration, they encountered a column of cells that all seemed to respond best to vertically oriented stimuli. And as they rotated their bar of light away from vertical, the responses got weaker and weaker, even though they were centered in the middle of the visual receptive field. So this defines what we consider to be the property called orientation selectivity. Now, not shown on this slide is a related property called direction selectivity, and that is that many of these neurons are going to respond to a bar of light that are moving just in one direction, but not in the opposite direction. Or they may at least have a strong preference for motion in one direction or the other. That we call direction preference. So most neurons in the primary visual cortex exhibit these two properties together of orientation preference and direction preference. More recently, newer methods have been developed to complement what can still be done very well with a microelectrode. These are results that I showed you previously in the course using a method called optical imaging of intrinsic signals. And this is a method that requires exposing the visual cortex to light and measuring the amount of light that's absorbed and the amount of light that's reflected. And what we know is that as regions of the brain become active, they increase their neural activity, which consumes more oxygen and induces a hemodynamic response, increasing local blood flow to those regions of the brain that have increased activity. Well, with this method, it's possible then to map out the local regions of the brain down to a few hundred microns that become active. And what's being represented here is the response to a bit of the visual cortex to a set of visual stimuli that vary in the orientation of the elements of that visual stimulus. And what we see when we drive the visual cortex with an oriented stimulus is a series of domains or columns or stripes that become active. And the columns that are driven by, let's say, a horizontal stimulus are precisely complementary to those columns that might be driven, say, by a vertical stimulus. And this complementary spatial location in the visual cortex can be represented by simple image processing methods where, let's say, a horizontal image is subtracted from a vertical image. And when you do that, you get something that looks like this, where now the dark regions are those that are driven by the vertical stimulus, and the white regions are those that are driven preferentially by the horizontal stimulus. And as I think you can tell, these regions perfectly interdigitate. Well, this experiment can be done over and over again for all kinds of combinations of visual stimuli, and we can represent this in a highly dynamic way. So what we're looking at here is an animation constructed by these images that represent the more high contrast black and white view of these interdigitating columns. And what I've done here is I'm showing you representations of the visual cortex from 3 different model species that we've used in the lab. And what I hope that you appreciate from this is really a couple of things. One is the smooth, continuous way that these columns that are driven specifically by given orientations are laid out across the brain. In fact, I think one can even identify that there seem to be regions where these columns seem to circle around one another in what almost look like to be circular points around which there is rotation. Well, of course, there's nothing really moving in the brain. Rather, the dynamic representation of activity is what we see in this animation here. And what this demonstrates is that there are adjacent regions of the brain that are representing slightly displaced orientations of visual stimuli. Well, at least for me anyway, this display can be a bit mesmerizing, especially if one reflects on the architecture of the circuitry of the visual cortex that accounts for these columnar patterns of activity and how they relate to one another as a visual stimulus is changed in a smooth and continuous fashion. Well, let's return to the more static display so we can begin to use this columnar system in the brain as a model system for probing the influence of experience in early life. So I'd like to just mention a couple of conventions because we'll want to use these in the experiments that are designed to assess the role of experience in shaping the circuits that give rise to these columnar patterns. You'll notice that the column that I have here to the left is labeled response. And so what we're looking at here is simply the activity in the brain that represents the presence of the visual stimulus. And to the right, where we have these beautiful dark and light interdigitating domains, we call this selectivity. This is now the result of subtracting one image from another. So the degree to which we see dark and light regions in this difference image, as we call it, is an index then of the selectivity of the cortical response. That is, if the cortex responded uniformly across all columns to the same visual angles, then when we subtract the two, we would expect to see just uniform gray. But the fact that we see these nice high contrast images of dark and light bands and stripes interdigitating with one another indicates that there is a high degree of selectivity where interdigitating columns respond quite strongly to their preferred orientation, and the fall off in activity is rather steep as we move away from their preferred orientation. Well, these data can be combined in different ways, and one way that we like to combine them is via an image processing routine called vector summation that allows us to assign a color to every pixel in the visual cortex. And the color of that pixel is now indicative of what is the best orientation that drives that location in the visual space map. And so what we find in the color code down below is a representation of orientation preference. And the intensity of the color tells us something about selectivity. So for example, where we see these nice intensely red regions, these are areas that responded best and quite selectively to horizontal stimuli. And where we see these intensely blue regions, these are columns or bands of cortex that responded best to near vertical orientations. Well, let's dive into this visual cortex and expand this representation, because I want to highlight the centers around which these columnar structures seem to organize. Now, I would draw your attention to these dark spots, which simply represent the limits of resolution of this method. But around these dark spots are arrayed all of the colors that define this color code for orientation preference. And what the distribution of these colors indicate is that there is a singularity around which circuits that represent all possible orientations are organized. And so we see this marvelous degree of organization repeated many, many times across the visual cortex. We call these point singularities pinwheel centers, and the complete structure that contains the columns that represent all possible orientations for a given set of overlapping receptive fields, we call those pinwheels. So the visual cortex then is organized with the circuits that take the shape of pinwheels, and these pinwheels repeat many, many times across the surface of the cortex. And in some work that I was privileged to participate in with some collaborators who are theoreticians and computational neuroscientists, what we were able to do is to examine a large set of orientation preference maps from different species. And we did a fairly simple analytical procedure, although it was not trivial to perform this. What we did was we simply assessed the density of pinwheels in the visual cortex. And when we plotted the density of pinwheels in units of area of measurement and related that to the overall size of the pinwheel, something that we call the hypercolumn size, what we found is that all the data points seem to follow along a single line. And when we simply divided these two axes from one another, that allowed us to generate a dimensionless expression called pinwheel density. What we found was that now all of these data points fall along a horizontal line. Well, what was especially exciting about that is that it suggests that there may be some kind of universal principle that describes the way this circuitry organizes in development. And working with some computational scientists, it was easy to get excited about exactly what this value was representing. What we did was we measured a very large sample and discovered that the average pinwheel density across our sample was, well, 3.14, which is statistically identical to the mathematical constant pi. In fact, our confidence interval, shown in parentheses here, indicates that pi is within our confidence limit of our ability to make this measurement in real brains using relatively imperfect experimental methods. So what should it mean that circuits in the visual cortex organized to form pinwheels, and the density of those pinwheels evidently is pi across a broad range of mammalian species? Well, we think this is a signature of self-organization. And some modeling experiments that ourselves and others have done indicate that there are really two important factors that account for the self-organization of these networks. One important factor is that these networks are dynamical systems, and they're subject to activity-dependent plasticity. Another important factor is that these networks grow out long-ranging axonal connections that are able to bind together the activity of different cortical columns that develop similar preferences. We think that these two factors together help to explain why the visual cortex organizes itself into these pinwheel formations with a density of pi.